FirePlex HT assays are read out on high-content images. The TIFFs generated by the imager are then analyzed and decoded by our integrated, free-of-charge FirePlex Analysis Workbench software. We recommend first running the FirePlex HT Imager Setup Kit to validate that your specific high-content imager will be compatible with our assays. FirePlex HT immunoassays are validated for use with numerous high-content images, including PerkinElmer Opera Phoenix, Molecular Devices Image Express, and GE Incell 2200 and 6000. The required specifications are detailed in the FirePlex HT protocol booklet. Included in each FirePlex HT kit are all of the assay reagents required for your experiment. This includes the FirePlex particle mix comprising your analytes of interest, a 384-well imaging plate compatible with high-content imager readout, and ancillary buffers and reagents. Upon receiving your kit, please unbox and store the reagents at the storage temperatures indicated on each file. The storage temperature of each component is also provided in the FirePlex HT protocol booklet. Please take extra care to note the storage temperatures of reagents before and after preparation may vary. For example, lyophilized protein standard mixes may be stored at -20 C before use, but should be aliquoted and stored at -80 C after resuspension. In addition to the materials supplied in your FirePlex HT kit, you'll also require access to some basic laboratory equipment and instrumentation. Please refer to the protocol booklet for the required materials that are not supplied. Protein standards should be prepared immediately before use with the FirePlex HT assay. The standards supplied in your FirePlex HT kit are for use with that specific FirePlex HT panel and should not be interchanged. To prepare your standard curve, dilute the lyophilized protein standard mix or mixes according to the instructions provided in the FirePlex HT protocol booklet. Take extra care to switch tips when preparing each dilution and mix thoroughly by pipetting up and down and vortexing. It is recommended to run the standard curve in duplicate, including 4 blank wells. This will ensure a more robust data analysis of your biological samples. After preparing your standard curve, place the tubes on ice until you're ready to set up your assay plate. Note that any remaining protein standard mix stock solution should be aliquoted and stored at -80 C. It is not recommended to store any of the remaining diluted protein standard mix. FirePlex HT immunoassays are compatible with plasma, serum, and cell culture supernatant samples. Day 1 of the FirePlex HT assay workflow entails reagent preparation and assay setup, followed by overnight incubation. Begin by preparing 1x dilutions of the FirePlex particle mix and FirePlex detector antibody mix using the provided reagent diluent. These two components are then combined at equal volumes to create the 1x reaction mix. Ensure that particles are resuspended and all reagents are thoroughly mixed before pipetting and during the preparation of the 1x reagent mix. The corresponding storage temperature of reagents may change after preparation. Please refer to the protocol booklet for instructions on reagent storage following preparation. Please note that some FirePlex HT reagents are light sensitive and should always be protected from light. These reagents are indicated with an asterisk in the table provided in the Materials Supplied section of the protocol booklet. Also, assays incubations should be carried out in the dark, either by using the imaging plate lid supplied in your FirePlex HT kit box or an opaque plate seal. FirePlex HT assays are most efficiently run as full plates, but if you do find yourself running fewer samples, refer to the table provided in the Assay Procedure section of the protocol booklet to adjust volumes as necessary. We've also provided an extra standard protein mix to ensure that multiple experiments can be run with each kit. We recommend using liquid handling instruments for reagent and sample dispensing when running FirePlex HT immunoassays. The FirePlex Field Application Scientist team can provide support on how to set up your liquid handling instrument for the best results. Refer to the protocol booklet to find the email address to contact our scientist team. When dispensing the reaction mix to each plate, ensure that the reaction mix is vortexed thoroughly to avoid particle settling. Samples can be added to your assay plate. Samples should be pre-diluted at the required dilution, using the sample diluent supplied in the FirePlex HT kit. Please refer to the protocol booklet for further instructions on sample dilution. We recommend that each sample is assayed at least in duplicate measurements. Remember, you may need to run multiple dilutions if analytes of interest differ largely in their abundance within a sample. When adjusting for dilution in your final analysis, make sure to account for the additional 1 to 2 dilution that occurs in the well. Upon adding all reagents and samples to the FirePlex HT assay plate, ensure that you seal the assay plate using either the supplied adhesive plate seal or an opaque plate seal. This will prevent sample spillage or loss during the overnight incubation or in the rare instance when a plate is dropped. Day 1 of the FirePlex HT assay workflow concludes by incubating assay plates overnight at room temperature while shaking on an orbital shaker. Refer to the FirePlex HT protocol for detailed instructions. Be aware that FirePlex HT immunoassays are light sensitive. Please take care and ensure that assay plates are incubated in the dark using either the supplied imaging plate lid or an opaque plate seal. Day 2 of the FirePlex HT assay protocol entails the addition of the 1X imaging dye and data acquisition using a high-content imager. The FirePlex HT imaging dye is used to quench any assay background. To prepare the 1X imaging dye, please refer to the protocol booklet for more details. Please note that following the overnight incubation, condensation may have built upon the plate seal. This can be removed by briefly centrifuging the assay plate. Gently remove the plate seal and add 10 microliters of imaging dye to each well. If you do not use all of the imaging dye that was prepared, it can be stored at room temperature in the dark for up to 1 week. Carefully handle the imaging dye, as any unwanted spillovers may stain bench tops and some plastics permanently. Make sure to store any imaging dye leftovers in the dark. The addition of the imaging dye can be done manually using a multi-channel pipette designed for 384 well plates. For example, Integra electronic pipettes work very well for this process. This step can also be carried out using a liquid handling instrument. After the dye is added, cover the plate with a fresh adhesive plate seal and incubate for 20 minutes at room temperature on the orbital shaker. Assay plates are now ready for imaging. Gently wipe off the bottom of the plate with a Kim wipe to remove any dust. The plate can also be gently vortexed at the corners to ensure that particles are evenly dispersed along the bottom surface of the well. Place the plate in the imager and begin the image acquisition using the instruction provided in the protocol booklet. We strongly recommend that users store plates in the dark at 4C following image acquisition. If necessary, plates can be rescanned for an additional 12 hours. The TIFF files generated by the high-content imager should be exported according to the instructions provided in the protocol booklet of the FirePlex HT Imager Setup Kit, or available directly via our Field Applications team. Images can then be imported into the FirePlex Analysis Workbench for analysis and analyte quantitation. From the supplied TIFFs, the FirePlex Analysis Workbench generates standard curves and quantitates of each analyte in a given sample. Data can be exported in CSV or XLSX format to be compatible for use with other data analysis programs such as GraphPad and GeneData.